Hello everyone. In this tutorial, today we will learn how we can download the genome FASTA file and also the gene annotation file, which is commonly in a GTF file format. So genomes and those annotation can be downloaded from different sources. Today we will show the demonstration how we can download those files from ENSEMBL database. So let's go to ENSEMBL website, download ENSEMBL genomes. So let's go to the FTP download website, and if we go down, we'll be able to see different genomes: human, mouse, zebrafish. So the ENSEMBL website has many different genomes which we can explore going through this website. If we open the home page, it will show all the genomes that are available in ENSEMBL, but today we are interested on the mice genome, so I will show how we can download the mouse genome. So here the genome name and DNA FASTA, cDNA FASTA, CDS, all kinds of sequences are linked here, and variants file, regulation file, bigwig, BAM, and different files. So let's click the genome file, the DNA FASTA, and this is taking us to the FTP server where it is saying the release is 115, and we can see there are different files. So it's important to note that there are different versions of the genome, such as it is telling that GRCM39. If we want to use the genome version 39, we can download those files. Those files are separated by different chromosomes. Also, there should have a file that has all the chromosomes together. So this is the primary assembly file. So this is the file we can download directly from ENSEMBL, and this will contain all the chromosomes. So there are options that we can download chromosomes separately. And let's go back to the directory in case we would like to download different versions of the genome. So here in the pub location, we will find all different releases. Current release is 115. So the genome here in the current version is 39. So let's go back to previous releases such as 102. And go to FASTA. We can find the mice genome, and you can see a difference here. In the release 102, the genome version was different, that is GRCM38. On the other hand, the release 115 had a different genome, which is 39. So this is very important to note that genome versions are updating. So we need to use a consistent genome version. If we use 38 in our analysis, we need to be consistent throughout our analysis. We cannot change or swap genomes for a given project. So today we will use this HG38, and I will show how we can download those data using bash script. So the primary assembly file that contains all the chromosomes, and we will use bash commands to download this file. So let's go back couple of steps. We would like to download the GTF file which contains the gene annotation, and we can see for each release there are different files. We can download bat file, gene bank format, GFF format, GTF format. So for gene annotation today we will look into the GTF file. And for the same release we need to be consistent with the release as well, because in different releases the gene annotation may change. So from here we will download this file that contains all the gene annotation. So always it is important to read the readme file. In each directory there is a readme file where you can find the details about each files, what those files means, what the data in those GTF files. For example, it is explaining the different values that are stored in a GTF file, gene ID, gene version, name, so this is always important to look into the readme file. So let's go back to the coding part, where I will show how we can use the bash commands to download all the files and do some changes and create some downstream files so that we can integrate those files into our bioinformatic analysis. To start the download, let's go to the primary assembly file and right click and copy link address. So this will copy the entire path from where we can download this file. So let's use curl command to download this file. So it copied the entire link. Now if we press enter, this will start downloading. So for Linux based system we can use also the wget command to download this file from the link. Let's wait for a couple of minutes because the genome size is around 2.6 GB. So the file has been downloaded. We can see the whole file is compressed and it is around 769 megabyte in size. I would like to show one thing about this assembly file. If we can open this file, this is a big file, so it's difficult to open. Just I wanna show one line. Sorry, my laptop is not good enough to open this file. Let's unzip this compressed file. And now we can see the file size is 2.6 GB. Let's try to open the header. So this is the first line of this file, which is showing the chromosome number and some description. So this is important to note. The chromosome numbers are indicated by numbers. In ENSEMBL by default they have the chromosome numbers such as 1, 2, 3, 4, 5. There is another convention to write the chromosome names by adding CHR, CHR1, CHR2, CHR3. So you can follow either of the conventions, but you need to be consistent in using either numbers or numbers with CHR character. Personally, I would like to use the chromosome names that starts with CHR, such as CHR1, CHR2, but either one you can use, but be consistent. So now we will download the GTF file using the same approach. We are going to the ENSEMBL website and right click, copy the link address. This will copy the entire link and we can download the GTF file. Again, I will unzip. Yes, this is the GTF file, and the first column is the chromosome number. So it is 1. So we need to keep going to see the chromosome number 2, but there is no CHR character. So again, we can use this reference file that has numbers for chromosomes. Also, we can use this TTF file. It has only the numbers to indicate the chromosomes. So these are consistent. But if you prefer to add CHR character, I have wrote down the code that adds the CHR character in front of every chromosomes. Now I am showing the entire script, how I can automatically download and add CHR characters before the chromosome numbers, and also how we can create the chromosome size files. This is a tab separated file that has chromosome name, and in the second column it has the size of the chromosome. On this chromosome sizes file, sometimes it's useful to use certain bioinformatics pipeline. And these are the steps showing how can we download GTF file and add CHR character. Then another thing I would like to do is convert the GTF file into BAT file. BAT file is sometimes very useful to intersect genomic coordinates and use in some tools such as BAT tools. So this has different applications when you convert a file into BAT file. So you can use this script to create a BAT file for all the genes. You can see I have selected the genes only from the GTF file. GTF file also contains the transcripts and other information, but this script only takes the all gene information and store this gene information into a BAT file. This section is particularly important for epigenetic analysis, such as if someone wants to look into the promoter region around the transcription start site, then this script will generate the windows around the transcription start site. For example, this chunk of the code will take the gene information, gene coordinates from this BAT file, and then will extract the window that is 1 kb upstream and 500 base pair downstream of the transcription start site for all the genes. And this is the same script but for 2 kb windows, that is 2 kb upstream and 2 kb downstream of the transcription start site. Also similarly for 5 kb windows. These scripts will generate the windows around the transcription start site for all the genes, but sometimes we are interested to know only the protein coding genes and their promoter windows. So using this command, we can actually separate the protein coding genes from all the gene sets. So let's run this script. If we run this script bash download, this script is here, then this will start download and processing and will generate all the files that you need. So I wrote an entire blog on this tutorial. Also, I have uploaded the scripts on my website, Bioinformatics Tutorials. Please try to download the data using this script. If you have any problem, please let me know. Also, this is important. Try by yourself to download different versions of the genomes or annotation files, or download the genome for like human or any other organism.